Hello, everyone. My name is Hugh Chu. I am an assistant professor in the Department of Pathology here at UC San Diego. And today I will be telling you about a story from our group where we examine how host glycans shape early life gut colonization and immunity. As many of you may know already, in humans, colonization begins at birth, where the infant is exposed to microbes during the delivery process, either via vaginal or C-section delivery. And over time, their microbial community becomes more complex, increasing both in microbial load and diversity. Now, in the last 10 to 15 years or so, microbiome association studies have described a link between the infant microbiome and the relative risk of developing immune or metabolic disorders later on in life. This includes allergies, asthma, atopic dermatitis, among others. And so the establishment of this early life microbiome during this critical developmental window can really have a profound impact on our health well into adulthood. And while we know what bacterial species are prevalent during early life, the molecular mechanisms that enable commensal bacteria to stably colonize the infant gut is much less well understood. And so in our lab, we leverage the model commensal bacteria, Bacteroides fragilis, to study the colonization program during early life. Now, what we do know are the many factors that shape the infant microbiome. I'll only touch on a couple of these as it relates to my talk today. But as I mentioned already, the mode of delivery can have a profound impact on the composition of the infant microbiome. We know that vaginally delivered infants are dominated by strains that are coming from mom, whereas in C-section delivered infants, their microbiota are dominated by strains that you would find in the environment, typically in hospital settings. The mode of delivery, as you can imagine, can also have a significant impact on the infant microbiome, whether the baby is feeding exclusively on mom's milk versus formula feeding or a combination of the two. And of course, mom's microbiome, both intestinal, vaginal, and skin microbiota, has a significant role in shaping the composition of the infant microbiome. And in fact, work by Marianne Yassur's group examined which bacterial strains are the most maternally transmitted in human infants. And despite the fact that infants are dominated by bifidobacterium during the first few months of life, their group actually found that Bacteroides species are actually the most maternally transmitted strains in humans. They found Bacteroides species as early as the first day of life, and they can track that these strains are identical to the ones that are found in their mothers. And so we were quite intrigued um, to see this finding. And we were curious as to whether there is a specific colonization program that allows Bacteroides to be so successfully transmitted from mom to infant and stably colonize the infant gut. And as I mentioned earlier, the consumption of human milk also has a significant effect on the infant microbiome. As many of you may know already, human milk contains over 200 different structures of human milk oligosaccharides. And while the infant themselves cannot metabolize these sugars, select gut bacteria such as bifidobacterium and Bacteroides species can actually break down these sugars as a carbon source. And so the paradigm was that the consumption of these human milk oligosaccharides via milk promotes the colonization of keystone species such as Bacteroides fragilis. But we actually hypothesized that perhaps um, human milk oligosaccharide metabolism can trigger a specific colonization program in commensal bacteria such as Bacteroides fragilis. And so to explore that, we collaborated with Lars Bodie and his group. And one of the first experiments we did was to take a collection of Bacteroides species and we grew it on human milk oligosaccharides. These are pools of human milk oligosaccharides from human donor milk as the sole carbon source. And hopefully, what you can appreciate is that there is a group of Bacteroides species that are able to use HMOs as a sole carbon source, and a collection of other strains that are unable to use HMOs as a sole carbon source. And for our studies, we primarily focus on Bacteroides fragilis as it is the most proficient at metabolizing and using human milk oligosaccharides. And so we tracked which specific HMOs Bacteroides fragilis preferentially uses um, over the course of 48 hours. And something that Lars and his group observed early on is that Bacteroides fragilis appears to prefer sialidated human milk oligosaccharides. And this corroborated some of our findings when we performed comparative proteomics of Bacteroides fragilis during different phases of growth. And so just to orient you, we looked at early growth on HMOs, mid log, and then also late log, early stationary growth. And what we observed in this data set is the presence of NANH in all three of these growth phases being significantly upregulated compared to growth of Bacteroides fragilis in a glucose control group. And NANH is a sialidase, GH33, that is enclosed in a locus that we believe to be important for human milk oligosaccharide utilization. There's a couple different cis-CD pairs that are important for sialic acid utilization. But in combination with the NANH, we also see the NAN operon and the DON operon being upregulated during early and mid growth. The NAN operon is important as it has the sialic acid transporter, and the DON operon is important, as the name suggests, for N-glycan utilization. And so we sought to see what the role of sialidase may be in growth on HMOs. And so we generated a knockout, an isogenic mutant of NANH in Bacteroides fragilis, and looked at its growth first on a nutrient-rich media, which there was no growth defect in the mutant when we compared it with the wild-type Bacteroides fragilis. But when we grow exclusively on pooled HMOs, there is a significant growth defect in the NANH mutant when we compared it with Bacteroides fragilis wild type. If we fractionate the pooled HMOs and separate out the neutral HMOs from the acidic, when we grow them exclusively on the neutral HMOs, these are the HMOs that do not contain any sialic acid. There's no difference in growth between the wild type and the NANH mutant. Whereas growth in the acidic HMOs, these are the HMOs that contain sialic acid, there is a significant growth defect in the NANH isogenic mutant compared to the wild type Bacteroides fragilis, indicating that the sialidase is required for fragilis growth on HMOs, specifically sialidated HMOs. Now, turning back to our comparative proteomic data, when we looked in the late growth phase, instead of some of the NAN and the DON operon proteins, we actually see an upregulation of these CCF proteins. And CCF stands for commensal colonization factor. And several years ago, CCF was described to be important for colonizing the colonic crypts. And in these EM images on the left are mice monocolonized with wild-type Bacteroides fragilis. You can see there's many bacteria. The bacteria are replicating. They are quite close to the intestinal brush border here. Um, and in the knockout of the CCF, you can see much fewer bacteria in the colonic crypts and also fewer bacteria in total. You can see in the wild type, there's just herds of these bacteria, whereas in the CCF, we have this lone bacterium in the mouse crypt, indicating the important role of this locus in commensal colonization. And so this actually led us to hypothesize that perhaps HMO metabolism turns on the CCF loci that is important for stable colonization in the mammalian gut. And to us, this made sense because if you compare the structure of the HMO glycan with the mucin O glycan, there's actually very similar, actually identical structures where we see NANH could potentially cleave the alpha 2,3 and the alpha 2,6 sialic acid that you see in both 3SL and 6SL in human milk oligosaccharide. And so we next look at colonization between the wild type bacteria and the NANH mutant. And in this experiment, we co-colonize the two strains of bacteria at a 1-to-1 ratio in germ-free black 6 mice. And we tracked the colonization of these two strains over the course of a month. And we can see a colonization defect of the NANH mutant as early as about a week after colonization. And this is perhaps more apparent when you look at the competitive index and see that after about a week, NANH becomes outcompeted by the wild-type Bacteroides fragilis, indicating that NANH is important for intestinal colonization. We next looked at the role of NANH in mediating colonization during early life. And here we took germ-free breeder pairs that are, again, co-colonized with the wild type and the NANH mutant. And we looked at the pups over the course of the first 2 weeks of life. And not surprisingly, we can see that the wild type Bacteroides fragilis has a competitive advantage over the NANH mutant in the adults. And this persists into the pups as well when these mice are feeding exclusively on mom's milk. These are just the stomachs of the 4, 7, 10, and 14-day-old mice and the composition where they're primarily contain 3SL and 6SL over the course of about 2 weeks. And so we see that NANH is important in mediating colonization during early life. We also wanted to see how NANH may play a role in competing against other Bacteroides species. And so here we co-colonize breeder pairs again, but this time competing either wild-type Bacteroides fragilis with Bacteroides theta iota micron, or the NANH mutant with theta. And hopefully you can see that in adults, theta actually dominates over Bacteroides fragilis, the wild-type strain. But in pups, you can see that wild-type actually outcompetes theta, perhaps due to its enhanced ability to metabolize human milk oligosaccharides. Now, if we perform this experiment in co-colonization studies with a NANH mutant, you can see that theta wins out in the adults as well as the pups. And so the lack of the single sialidase NANH is enough for Bacteroides fragilis to lose out to theta. during early life. And we see the similar pattern when we perform these studies in germ-free adult mice, where Fragilis is able to stably colonize and kind of coexist with theta over the course of 21 days, whereas the NANH mutant essentially loses out within the first week or so. We next wanted to look at the role of the NANH sialidase in resilience during antibiotic perturbations. As many of you know, um, many children before the age of 2 go through at least one course of antibiotics. And so we were curious whether NANH may be important in resilience during this kind of antibiotic course. And so here we decided to take a simple microbial community. This is a 9-member community together with wild type and NANH at a 1-to-1 ratio. And we allowed them to colonize a mouse for about 10 days before we added the antibiotic ciprofloxacin in the drinking water. And as you would expect, the wild type Bacteroides fragilis has a competitive advantage over the NANH. And after the course of antibiotics, all bacterial strains go down from the antibiotic treatment. And after we remove the Cipro from the drinking water, we wanted to see how both of these strains rebound, if at all. And what you can see is after 20 and 27 days, Bacteroides fragilis wild type rebounds back to kind of the levels that you would see before the antibiotic treatment, whereas the NANH mutant are at a pretty low level, almost undetectable relative to the wild type bacteria. And all of this was quantified by qPCR relative to total 16S. We also enumerated this by colony forming units by in the fecal pellets as well as in the mucus. And again, you can see that the wild type strain is much more resilient and can persist after antibiotic perturbation, much more so than the NANH mutant. Importantly, we wanted to see what are the immune outcomes in the wild type versus NANH colonized mice. And we can see that when we look at Treg populations, the NANH mutants have a significant reduction in FOXP3 Tregs and IL-10 production among these FOXP3 Tregs, but a reciprocal increase in pro-inflammatory Th17 responses, suggesting that perhaps the NANH sialidase is important in mediating immune tolerance in the infant gut. And so several questions that that comes up when we've presented this work in the past is whether sialylated human milk oligosaccharides can provide can be supplemented in infant formula to promote a healthier microbiome. And so as I alluded to at the beginning of the talk, babies that are born via vaginal delivery, born at full term, are fed via breastfeeding, their microbiota typically are dominated by Bacteroides species and Bifidobacterium, whereas infants that are born preterm or via C-section delivery or primarily formula fed, their microbiota are typically dominated by Enterobacteriaceae, and we see a reduction or sometimes a complete absence of Bacteroides species and Bifidobacterium that, as I mentioned, is associated with developing metabolic and immune mediated disorders later on in life. And so we wanted to test whether we can supplement with sialylated human milk oligosaccharides to push the microbial community closer to what you would typically see in a healthy microbiota. And so we performed these experiments in mice where we colonized germ-free mice with what is a 8-member preterm community. So these are strains that we isolated from 2 different babies from the NICU. They were born preterm. And as you can see, these are predominantly Enterobacteriaceae. But we added Bacteroides fragilis. This is, again, our model commensal bacteria that expresses NANH. Interestingly, none of the strains that were isolated from the preterm microbiota expresses the sialidase, but almost all of them, with the exception of Enterococcus, express the sialic acid transporter NANT. And so we performed the experiment where we colonized germ-free mice with this community. And we either gave the mice just regular autoclave drinking water or drinking water supplemented with the sialidated HMOs 3SL and 6SL. And we just performed this experiment over the course of 7 days. And we tracked the percentage of Bacteroides fragilis relative to total 16S. And you can see that when the mice are drinking just control water, Bacteroides fragilis is roughly about anywhere between 15 to 20% of the community. However, when we supplement their drinking water with 3 and 6SL, you can see that after about 7 days, Bacteroides fragilis is actually the dominant member of this preterm community, suggesting that perhaps supplementation with sialidated HMOs with a beneficial bacteria that expresses a sialidase, such as Bacteroides fragilis, may be able to restore the microbial community to something resembling what you would typically see in a healthy full-term infant. Importantly, I want to stress that NANH is actually a core gene found in all Bacteroides fragilis strains. Our work was primarily with the type strain NCTC9343, but we did isolate 2 Bacteroides fragilis strains from 2 infants that were breastfed. And you can see that both of these strains are able to grow just as well, if not slightly better, than our type strain, suggesting that perhaps these findings are translatable to Bacteroides fragilis strains in all infants. And I wanted to actually also highlight the study that was published a couple years ago from Jeff Gordon's group, where they looked at sialidated milk oligosaccharide supplementation in a microbial community that came from malnourished infants. And what they showed is that supplementation with sialidated bovine milk oligosaccharides were able to promote the growth. This is as measured by lean mass gain, significantly more so than the inulin control. And when they looked at what bacterial strains were the most responsive to this sialidated BMO control, what they found was actually that it was Bacteroides fragilis transcript, namely these SUS-Cs and SUS-Ds that are part of the HMO polyutilization loci that we described in our study, suggesting that sialidated milk glycans may be an effective supplementation also for infants that are malnourished, to promote not just colonization, immune health, but also growth of these malnourished infants. And I also want to leave you with the notion that sialidase and sialic acid utilization is actually quite common in the human gut microbiota. There's more than about 80 different bacterial genera that express either a sialidase or a sialic acid transporter in the human gut microbiota, suggesting that perhaps this is a mechanism that other commensal bacteria may utilize to stably colonize the human gut. And so hopefully I've showed you today that human milk indeed promotes stable colonization of a commensal bacterium, Bacteroides fragilis, that can potentially provide lifelong benefits to the host. And we are now exploring additional immune readouts to see whether either supplementation with salidated HMOs in combination with Bacteroides fragilis can promote immune tolerance and protect against things like infection and inflammatory disorders in various mouse models. And so I just want to thank the folks in my lab that contributed to this work. This work also could not have been done without our very active collaboration with Lars and his group. And our proteomic studies were performed in collaboration with David Gonzalez and his lab, and the funding that made this work possible. Thank you.